Hello, I'm way down here. Hello, I'm here now. No, I'm way down here now. What is your MERVL gag? And do you have one? This is interesting. So this just recently came out. This isn't even that old. Came out first of the year or the end of last year, right at the end, maybe. Here's some more. Well, they found it 2022, but something has happened. Maybe I just found it recently. I should redo this, but I'm not going to. Yes, so here they've been working on it, but we have some new information coming out. So I'm going to go to a very easy to read instead of doing what I normally do and read one of the papers. You can clearly see there's lots of papers. Let me show you how these work. So you can, this is cited by 24 people. So this is a paper because it's cited. It tells me that it's a paper there. This would be more of an article. And then you get a paper. This is cited by one. And this one is something you can buy if that shouldn't bother you. It's a US $99 up to $399. So you can actually buy this, what I'm about to share with you. There's another cited by 77 people from 2014. So that one being an older one, it'll get cited, then they'll run theirs, and then they'll cite it, and they'll run theirs. Anyway, you end up with something here is $315. It's in stock. You can buy one now. And of course, Berkeley's in on it. And protein serves a yet undefined role in fate potency. What does that mean? So I'm going to read to you what this is in normal people form. This is from Technology Networks Genomics Research. And so this is in like a periodical for smart people. So what they have figured out is this virus guides embryo development after infecting primitive organisms millions of years ago. I've recorded or shared or will be something along these lines as well. This is very interesting what I unlocked here, or what I discovered here. So an ancient viral remnants buried in the genome contribute to a, quote, smooth transition in embryonic development. So what does that mean? Well, that's going to mean a lot in epigenetics. So ancient viral fragments are far from junk. Millions of years ago, ancient retroviruses infected some of the world's earliest organisms. Oh yeah, we did a video about it. That's right, it's very interesting. While most viruses infect their host, replicate their DNA, and then leave, retroviruses stick around. They reverse transcribe their RNA genome into DNA, and under certain conditions, this DNA can integrate into the host's genome. When this integration occurs in germ cells, a sperm or an egg cell, the viral genetic material can be passed on to subsequent generations. Endogenous retroviruses, or ERVs, are remnants of ancient retroviral infections that have permanently integrated into the host organism's genome. For a long time, ERVs were thrust under the umbrella category of junk DNA. Now, what is junk DNA? This is the term that is often used to describe DNA that does not encode protein or lacks a clear functional role in the organism. So we're not sure what it does yet. As genomic tools advance over recent decades, it became increasingly clear that junk DNA is not all junk. It might even be treasure. Obviously, it isn't. None of it would be junk. There would never be any reason. Researchers have discovered important functions for viral remnants in the genome, such as gene expression regulation, genome stability, maintenance of chromosome structure, and other cellular processes. It's plausible that these viruses actually laid the foundation for many of the traits that make us human. And in their last research, they demonstrated how EVRs play a role in embryonic development, particularly during the essential transition from totipotency to plural potency, the study says. Now remember, you can buy this now, so that should scare you. The journey for $300, I can buy it. The journey from totipotent to pluripotent. Embryonic development is a complex process that involves many stages of cell division and differentiation. Our paper describes research as the role of EVRs in the development of mouse embryos, specifically in the transition from totipotency to pluripotency, which occurs shortly after fertilization. In the context of embryonic development, totipotent cells, so hard for me to say, can give rise to all the cells needed to form a complete organism, including embryonic cells and extra embryonic cells. And pluripotency is a more specialized stage than the other. These cells have the ability to differentiate into any cell type in the organism, but cannot form extra embryonic tissue such as the placenta. These cells are pivotal for the early stages of embryonic development because they eventually give rise to various cell layers, tissues, and organs of the organism. And this guy's group has spent a lot of time studying a complex called the unconventional pre-folding RPB5 intraceptor, or URI, encoded with the URI gene. Its function is largely unknown, but in worm and fruit fly studies, deletion of URI result in the death of the embryo. So it doesn't survive. In this study, the researchers utilize a suite of techniques to explore why, including genetically engineering mouse models, molecular biochemistry techniques, and bioinformation studies. So bioinformatics is not only complements our experimental work by handling vast data sets, but also allows us to extract meaningful insights from complex biological information. Its growing influence in our research enhances our ability to analyze, interpret, and derive valuable conclusions from the wealth of molecular data we generate. The synergy of these diverse approaches ensures a comprehensive and nuanced understanding of the biological processes under investigation. Ancient retroviruses help to modulate gene that is critical for the pluripotency. One role of URI is to enable other molecules that are required for pluripotency to kick into action. Without the URI, this doesn't happen, and that's where the retroviruses come into play. They found that an endogenous retrovirus protein known as the MERVL GAG helps to modulate our URI. During the totipotency phase, when there are only 2 cells in the oocyte, the expression of the viral protein MERVL GAG is high. High expression of the vital protein corresponds with fewer pluripotency factors. The MERVL GAG binds to URI and prevents it from acting. When the expression of MERVL decreases, pluripotency emerges. The URI binds and shields pluripotency factors OCT4 and SOX2 from proteasome degradation. It's that proteasome degradation. While the endogenous retrovirus protein MERVL displaces URI from pluripotent factor interaction, causing their degradation. High expression of the viral protein corresponds to a low presence of pluripotent factors as it interacts with URI, and pluripotency factors are no longer protected by degradation. As these factors degrade, the totipotency is favored. And quote, when the expression of MERVL decreases, URI is released from the interaction with MERVL and can interact with the pluripotency factors to protect them from the degradation. The research presents a completely novel role for EVRs and reveals a new mechanism by which they directly control the transition from toti to plurally. Plurally. I don't know why I keep trying to put an L there. Pluri. Endogenous retroviruses as support. You might have to put up with me not being able to say these things, but find another channel that reads this shit and shares it, and we'll all go over there. Endogenous retroviruses support a, quote, smooth progression of early embryonic development. Our findings reveal the symbiotic co-evolution of EVRs with their host cells, ensuring a smooth and timely progression of early embryonic development. This co-evolution allows sufficient time for the embryo to adapt and coordinate the seamless transition from toti to pluri, and the specification of cell lineage during embryonic development. The study was conducted in mice, which means its findings are not yet applicable to humans. We're doing it in underground bunkers as we speak. But if the mechanism discovered in this study also exists in humans, what implications could it have? Since it refers to the ability of a cell to develop into any cell type as well as into all tissue and organs of the complete organism. This URI inhibitors could induce this state, opening new avenues of research in the field of developmental biology. And there it is, regenerative medicine. So we're talking about designer babies and living forever, including the creation of artificial embryos. What the hell is an artificial embryo? Are we saying that we can make an embryo that is neither from sperm nor egg? Would that be a human being born without a soul, or a human-looking entity being born without? I mean, I don't know. I'm just saying, what does an artificial embryo mean? Therefore, the significance of human relevance becomes apparent, emphasizing the importance of developing URI inhibitors for further investigation in these fields, he concludes. I mean, this is just, I'm just going to copy this right here to put in the description. So anyway, that's some information there. And there's another, if you keep reading about this, literally what that says there, this is now opening them up, and you can buy it to practice on your mice for $100 to $399 from Epigen Tech. Will sell you a MERVL gag retrovirus-like particles. They de-repress and produce 3% of the transcribes mRNA at the 2-cell T2C stage. So you can just buy it, you know, there. Right after the expression is silenced. It has been found that this regulated pattern of MERVL expression overlapped with more than 100 C2 specific genes that have co-opted regular elements for the foreign retroviruses to begin. I just ask you, what really did we just experience? Was this a fear-mongering campaign, or was this a preparing I'm just preparing the operating room for the operation? I'm getting so many people to come to me and let me poke and prod them so that I can get more information and more information and more information. And then we all scream, you know, transparency, transparency, tell us what you're doing. So, you know, they just keep that keeps us safe when they say, you know, we're using it for this. What a sick world. I don't know what I just read, but it doesn't sound good to me. Anyway, I think I'd like to go back to my fairy tales again, where we weren't gagging our MERVLs.